Right now, your body is being attacked, not by a virus, not by a predator, by the universe itself. Every second you exist, cosmic radiation is tearing through your cells. Particles fired from dying stars billions of years ago are passing through your skull at this very moment. You don't feel them, you never do, but they're there, and they always have been. And the only reason you're still alive, the only reason any human being has ever survived for more than a few years on this planet, is because of a nearly invisible shield stretched around the entire Earth. A magnetic cocoon so enormous it dwarfs the planet itself. A field so powerful it deflects the solar wind, traps charged particles, and creates the auroras that glow at the poles like fire curtains. Take that shield away, and a human being lasts approximately 6 minutes before the radiation damage becomes irreversible. Welcome to deep space. This film is about a journey. Not the journey of a spacecraft, but the journey of a human body, your body, into an environment so hostile, so alien, so fundamentally incompatible with the chemistry that built you, that every single system inside you begins to fail the moment you leave Earth's protection. We're gonna go through it together, system by system, organ by organ, problem by problem, and along the way, we're going to meet the scientists, the astronauts, and the engineers who have spent their entire careers trying to solve what might be the most complex engineering problem in human history. Keeping a fragile bag of warm salt water alive in a void that wants it dead. This is the real reason no one can survive deep space. And the answer is stranger, darker, and more terrifying than you've probably imagined. In 1961, a young Soviet Air Force pilot named Yuri Gagarin strapped himself into a metal sphere barely the size of a small bathroom and became the first human being in history to see Earth from space. The flight lasted 108 minutes. When he landed, when he was found in a field in rural Russia, still in his orange pressure suit, waving at a confused farmer, he was described as looking slightly dazed. Grinning, but dazed. What the headlines didn't mention, what the Soviet state certainly didn't broadcast, was that his heart rate during re-entry had spiked dangerously, that his body had behaved in ways the scientists hadn't fully predicted, that even in 108 minutes, less than 2 hours, the human body had already begun to change in response to space. We talk about space exploration as if it's primarily an engineering problem. Build a rocket big enough, design a spacecraft sturdy enough, chart a course accurate enough. And yes, those are extraordinary challenges. But every engineer who has actually worked on human spaceflight will tell you the same thing. The hardest part isn't the rocket. The hardest part is you. The human body is not a machine. It's a biological system shaped by approximately 3.8 billion years of evolution on a single, very specific planet. Every organ, every cell, every molecule of you has been refined by an almost incomprehensible number of generations to function in one particular set of conditions, one particular gravitational pull, one particular atmospheric pressure, one particular cocktail of gases, one particular magnetic environment, one particular radiation level, one particular rhythm of light and dark. Change any of those conditions, and the body doesn't just struggle. It fights back. It adapts in the wrong direction. It begins, slowly, invisibly, relentlessly, to break down. The moment a spacecraft leaves low Earth orbit and heads into deep space, it crosses a threshold that no human being has ever crossed and returned from. It leaves the protection of Earth's magnetosphere, and at that point, the body is exposed to an environment that, in terms of its lethality, is unlike anything your biology was ever designed to handle. Let's start at the very beginning of that exposure. Let's start with what happens to you in the first few minutes after the magnetosphere disappears. Picture a bullet. Now, picture a bullet so small that it passes through solid steel without slowing down. A bullet that carries enough energy to break chemical bonds, shatter DNA strands, and trigger cancer in a single impact. Now, picture an infinite storm of those bullets, coming from every direction simultaneously, never stopping, never weakening, never once caring that you're in the way. That is galactic cosmic radiation. It comes from supernovae, from exploding stars throughout the Milky Way and beyond. The particles are accelerated to nearly the speed of light by the shock waves of these stellar explosions, and they flood the galaxy in every direction. They have been traveling for millions of years by the time they reach our solar system, and in most of the solar system, they hit you without interruption. On Earth, you are protected by two things. The first is the magnetosphere, that enormous magnetic bubble generated by Earth's molten iron core. It deflects a huge fraction of charged particles before they can reach the atmosphere. The second is the atmosphere itself, a column of gas roughly equivalent in shielding mass to 10 meters of water, which stops almost everything the magnetosphere doesn't catch. In low Earth orbit, where the International Space Station lives, you still have partial protection from the magnetosphere. Astronauts on the ISS receive roughly the equivalent radiation dose of a full-body CT scan every 5 days. That's significant. But it's manageable. The cumulative dose over a 6-month mission, while exceeding what any person on Earth would normally receive, falls within limits that NASA and its medical experts have determined to be acceptable. Not safe exactly, but acceptable, within the context of an informed adult choosing to accept a calculated risk. Beyond the magnetosphere, in true deep space, you have neither of those protections. The radiation dose increases by roughly a factor of 3 to 5. On a journey to Mars, which at its closest approach takes about 6 to 9 months one way, a crew would absorb somewhere between 300 and 700 millisieverts of radiation just from the transit. To put that in context, the current lifetime limit for NASA astronauts is somewhere around 600 millisieverts. A round trip to Mars, just the travel time, might use up that entire lifetime allowance, and the crew would still need to spend months on the Martian surface before making the return journey. And Mars transit is still relatively close. The further you go, the worse it gets. On a journey to the outer solar system, to Jupiter, to Saturn, to the Kuiper Belt, to the edge of the heliosphere, the transit times stretch into years. The radiation exposure becomes staggering, and at a certain distance from the Sun, even the solar wind's influence diminishes, and the galactic cosmic ray flux actually increases, no longer partially deflected by the solar magnetic field. But here is the part that keeps radiation physicists awake at night, that makes the problem qualitatively different from simply receiving too high a dose of X-rays. It's not just the total dose, it's the nature of the particles themselves. Galactic cosmic rays are high-energy heavy ions, particles like iron nuclei stripped of their electrons, moving at relativistic speeds, which is to say speeds approaching a significant fraction of the speed of light. When one of these iron nuclei hits a cell in your body, it doesn't damage the cell the way a photon from an X-ray machine does. It creates what physicists call a particle track, a line of total biological destruction running through every molecule in its path. It doesn't just nick a DNA strand. It doesn't just break one bond. It shatters everything along that track. DNA strands are cut in multiple places simultaneously. Proteins are destroyed. Cell membranes are disrupted. The damage is so complex and so concentrated that the cell's repair mechanisms, which evolved over billions of years to deal with conventional radiation damage, simply don't know what to do with it. A NASA-funded study published in 2018 found that mice exposed to heavy ion radiation experienced significant long-term cognitive impairment, measurable problems with memory, attention, and decision-making, even at doses lower than what a Mars crew would receive during transit. The neurons in the hippocampus, the brain region most critical for memory formation, showed structural damage. Dendritic spines, the tiny branches that connect neurons to each other across synapses, had been destroyed and not rebuilt. The neural architecture of memory had been physically damaged. That study caused a quiet panic in the human spaceflight community, because it suggested that the most dangerous thing about radiation in deep space might not be cancer. Cancer has a latency of years to decades. You might not know until long after the mission is over. The cognitive impairment showed up in the animal studies weeks after exposure. It suggested that the most dangerous thing about radiation in deep space might be that you arrive at your destination after months in the void and find that your mind doesn't quite work the way it used to, that your spatial reasoning is slower, that your memory is less reliable, that your risk assessment is subtly distorted. Charles Lemoli is a radiation biologist at the University of California, Irvine, who has spent a significant portion of his career studying exactly this problem. His lab has exposed rodents to simulated cosmic ray environments using particle accelerators to generate beams of ions similar to those found in the cosmic ray spectrum, and watched in detail what happens to their brains. What he found was consistent and disturbing. The animals showed increased anxiety-like behaviors, reduced cognitive flexibility, and significant deficits in spatial navigation. The ability to learn and remember the layout of an environment. They made more errors on maze tasks they had previously mastered. They learned new tasks more slowly. They showed behavioral changes consistent with elevated anxiety that persisted long after the radiation exposure ended. Think about what that means for an astronaut. You have spent 9 months traveling to Mars. You have absorbed a massive radiation dose, heavier than anything any human body was designed to absorb. And when you arrive, your spatial navigation is compromised, your anxiety is elevated, your reaction time is slightly slowed, and your cognitive processing has measurably declined. Now, you have to land a spacecraft on an alien planet, execute a complex surface mission, manage systems and experiments and emergencies, and eventually navigate home. The radiation isn't just a cancer risk you'll worry about after the mission. It's a cognitive impairment risk that affects your ability to complete the mission itself. The radiation problem has no perfect solution. Scientists have explored pharmacological approaches extensively, giving astronauts antioxidant compounds that mop up the free radicals generated by radiation hits. Some of these approaches show modest benefit in animal studies. Compounds like N-acetylcysteine, alpha-lipoic acid, melatonin, and certain plant-derived flavonoids have all been investigated as potential radioprotectors. Some show real, measurable effects in animal models at conventional radiation exposures. The problem is that heavy ion radiation injury, with its densely ionizing particle tracks, involves different damage mechanisms from the oxidative stress that these antioxidants are designed to address. The most devastating damage, the complex DNA breaks, the structural neuron damage, doesn't respond well to antioxidant treatment. Physical shielding helps, but only up to a point. Water is an excellent radiation shield for some types of radiation, because hydrogen nuclei are effective at absorbing and scattering energetic particles. This is why some spacecraft designs route the water supply through the walls of the sleeping compartments, creating a zone of reduced radiation for the many hours per day that crew members spend sleeping. But water is enormously heavy, and every kilogram you launch into space represents a real cost in fuel and mission capability. You cannot build a wall of water thick enough to stop galactic cosmic rays without making the spacecraft too heavy to launch with any currently existing rocket. Polyethylene, the plastic used in ordinary household bags, is actually a more effective cosmic ray shield per unit mass than steel. This is counterintuitive because we associate shielding with heavy, dense materials, but the physics favors hydrogen-rich materials for this type of radiation, and polyethylene has more hydrogen atoms per kilogram than almost any other solid material. Advanced spacecraft designs use polyethylene composite walls and structural elements specifically for this radiation shielding benefit. But even polyethylene walls several tens of centimeters thick cannot reduce the galactic cosmic ray dose to a level that any radiation safety standard would call acceptable. The highest energy cosmic rays, the ones carrying the most damage potential, simply pass through. There is a harder truth here that the space agencies rarely advertise prominently. No current or near-future spacecraft design can adequately shield its crew from galactic cosmic radiation on a multi-year deep space mission. The physics will not allow it at any practically launchable mass. Engineers can reduce the dose by meaningful amounts. They can optimize the shielding to protect the most sensitive organs, the bone marrow, the brain, the eyes, more than others. They can design mission architectures that minimize time in the highest radiation environments, but they cannot bring the dose down to a level that falls within any rational radiation safety standard. The crew will be significantly irradiated. The only question is exactly how much, and whether the specific individuals on the crew can tolerate that dose better or worse than average. The most promising long-term solution lies at the intersection of pharmacology and genetics. Researchers are beginning to identify specific gene variants that affect radiation sensitivity, that determine whether a given individual's DNA repair machinery is more or less effective, whether their cells are more or less prone to radiation-induced transformation, whether their nervous system is more or less vulnerable to heavy ion damage. If those variants could be identified reliably, crews could be selected partly on the basis of radiation resistance. More ambitiously, if the biological pathways that confer radiation resistance could be identified and pharmacologically targeted, a drug could potentially be developed that temporarily upregulates these protective mechanisms during the highest exposure phases of a mission. Some animal research suggests this is possible in principle, but translating it to a safe, effective human intervention is decades of work. For now, the radiation problem remains the deepest unsolved challenge of deep space human biology, and yet it's only the first of many. Let's talk about what happens to the body that is not being irradiated, but is simply floating. In April 2017, Scott Kelly gave a press conference. He'd just spent a year on the International Space Station, the longest continuous spaceflight in American history at the time, and journalists wanted to know what it had been like. Kelly smiled, answered their questions with the calm precision of a career test pilot. He looked healthy. He looked rested even. He fielded questions smoothly, cracked the occasional dry joke, gave every impression of a man in excellent health who had just completed an interesting professional assignment. And meanwhile, somewhere inside him, his bones were in the process of slowly rebuilding themselves from nearly a year of near-total weightlessness. His cardiovascular system was relearning how to manage blood pressure against gravity. The muscles that had spent 12 months barely needed were tentatively re-engaging with mechanical loads they had almost forgotten how to bear. His balance system was still recalibrating, sending slightly unreliable information about his orientation in space. Kelly looked fine, he sounded fine, but in a very real biological sense, he was not fine. He wouldn't be fine for months. Here is something that most people find deeply counterintuitive about the human body. It is not a fixed structure. It is a dynamic system in constant conversation with its environment. The density of your bones, the diameter and strength of your muscle fibers, the calibration of your cardiovascular output, the sensitivity settings of your inner ear's balance apparatus. All of these are continuously adjusted by your body based on the signals it receives from the world around it. Your skeleton today is not the same skeleton you had a year ago. Every bone in your body has been partially replaced over that period. As osteoclast cells broke down old bone tissue, and osteoblast cells laid down new calcium matrix in its place. And the most important signal driving, that constant process of maintenance and renewal, the one that maintains virtually every structural system in your body, is gravity. The simple, constant, downward pull that you have never in your life been without. Until you go to space. Without gravity, the human body begins to disassemble itself. The most dramatic example is bone density. In microgravity, bones lose density at a rate of approximately 1 to 2% per month in the most vulnerable regions, the hips, the femoral neck, and the lower lumbar spine. This happens because of a simple mechanical logic. On Earth, the skeleton is continuously loaded by gravity, compressed, bent, and tensioned during every moment of standing and walking. Those mechanical loads stimulate the osteoblasts to maintain bone density to keep laying down new calcium matrix. Remove the loads, and the signal to build bone disappears. The osteoclasts, the cells that break down old bone, keep doing their job. The osteoblasts slow down dramatically. The net result is a loss of bone mineral density at a rate that dwarfs even the most severe cases of age-related osteoporosis seen in elderly patients on Earth. After 6 months on the ISS, astronauts have typically lost enough bone density in the hip and spine that their skeletal profiles, measured by the DEXA scans performed routinely post-mission, resemble those of people 20 or 30 years older. After 12 months, the comparison becomes starker. And unlike muscle tissue, which begins rebuilding relatively quickly once exercise resumes and mechanical loading returns, bone recovery is slow. Studies following astronauts for years after long-duration missions show that some degree of bone density loss in the most affected regions persists for years. Some of that structural integrity may never fully come back. The implications for a Mars mission are direct and serious. An astronaut arriving at Mars after 6 to 9 months in microgravity transit, with significantly reduced bone mineral density in the weight-bearing regions of the skeleton, then immediately encounters a gravitational field approximately 1/3 as strong as Earth's. Mars gravity is much less than Earth gravity, but it is still enough to load a weakened skeleton. Stress fractures during physical activity on the surface, fractures during landing operations, fractures from the normal physical demands of suit work. All of these risks are substantially elevated in a skeleton that has been demineralized for months. A hip fracture in a pressurized suit on the surface of Mars, hundreds of millions of kilometers from any surgical facility, is not a recoverable situation. Current countermeasures focus primarily on exercise. ISS astronauts spend approximately 2 hours every single day performing prescribed exercise, using resistance equipment specially designed for microgravity, running on treadmills held against the belt by bungee cord attachment systems, pedaling stationary bikes with precise power output monitoring. This regimen has dramatically improved bone and muscle preservation compared to the early long-duration missions of the 1970s and 1980s, when Soviet cosmonauts returning from Mir sometimes couldn't walk unaided for weeks. But even the best current exercise protocols, using the best available equipment, do not fully prevent bone loss in the most vulnerable regions during missions of 6 months or longer. They slow it significantly. They do not stop it. The cardiovascular changes are in some ways more acutely dangerous. In microgravity, blood no longer pools in the legs and lower body the way it does on Earth. The hydrostatic pressure gradient that gravity normally creates, with higher blood pressure at the feet and lower pressure at the head, disappears entirely. Blood redistributes through the body more uniformly. The heart, which on Earth must pump against that pressure gradient to deliver blood upward to the brain, suddenly finds its job dramatically easier. It adapts. Over weeks and months in space, it reduces its output. Its walls thin slightly. Its chambers contract. The total blood volume falls as the kidneys, sensing the apparently elevated circulating volume, begin excreting the excess. The result is a heart that is perfectly adapted for space and dangerously maladapted for any return to gravity. astronauts arriving back on Earth after long-duration missions frequently experience orthostatic intolerance, an inability to stand without the blood pressure dropping so severely that consciousness is threatened. The heart, which has spent months not having to pump hard to send blood upward, suddenly faces the full challenge of Earth's gravity with a smaller, weaker, lower volume cardiovascular system. The blood falls into the legs. The brain, deprived of adequate perfusion, sends warning signals. The person sways, grays out, and collapses. This is manageable at a professional landing in Kazakhstan, surrounded by a flight surgeon and a medical team. It is not manageable at a landing on Mars, where the crew must operate independently and immediately. The cardiovascular impairment of landing day is one of the specific reasons why some Mars mission architectures include an orbital phase after the transit, a period of gradual readaptation to gravity before the actual landing, even though this adds complexity and cost to the mission. And then there's what happens to the pressure inside the skull, and what that does to the eyes. This is the discovery that shook the spaceflight medical community more than almost any other finding of the past 20 years, and it came almost entirely by accident. Astronaut John Phillips, who flew a 6-month ISS mission in 2005, came back to Earth and failed his post-flight vision exam. Not by a little. His visual acuity had dropped significantly enough to require corrective lenses he hadn't needed before. The medical team noted the finding, wondered about it, and moved on. Vision changes had been reported anecdotally before. They were assumed to be transient, perhaps related to the fluid shift. But over the following years, as NASA systematically began to look at vision data from all long-duration ISS missions, the pattern became impossible to ignore. Roughly 40% of long-duration astronauts, nearly half, were experiencing some form of vision change. Some were minor and recovered fully. Others were significant and partially permanent. And when the researchers began doing detailed imaging of the affected eyes, MRI scans, optical coherence tomography, fundus photography, what they found was deeply alarming. The backs of the eyes had changed shape. The optic nerve heads showed swelling. In severe cases, the choroid, the vascular layer at the back of the eye, showed folds, meaning the tissue had been physically deformed by some sustained force pressing against it from behind. That force was intracranial pressure. The cerebrospinal fluid that surrounds the brain and fills the space around the optic nerves had increased in pressure, pressed chronically against the back of each eye, and over months had physically remodeled the ocular anatomy. This was not a vision problem in the conventional ophthalmological sense. It was not a lens problem, or a corneal problem, or a retinal problem in isolation. It was the consequence of the brain pressing against the eyes. NASA named the condition Spaceflight-Associated Neuroocular Syndrome, SANS, and declared it one of the 5 highest priority medical risks of long-duration spaceflight. The designation was well earned. Unlike most of the other physiological risks of space, SANS causes structural changes. The eye is physically reshaped, and physical reshaping, unlike functional changes, does not reliably reverse when the cause is removed. Some astronauts who have experienced significant SANS have been permanently removed from flight status. Their vision did not come back to normal after returning to Earth. For a Mars mission lasting 2 to 3 years, with no possibility of early return, the implications are severe in a very specific way. If a crew member begins to experience progressive vision loss from worsening SANS during the mission, and the 6-month ISS data suggests that SANS worsens with mission duration, not stabilizes, there is no option for early return. There is no ophthalmologist on Mars. There is no ophthalmic surgical suite. The crew member will continue to lose vision. How rapidly and to what final degree of impairment, we cannot predict. We have no long-duration data. We have never sent anyone to Mars. We don't know what 3 years of elevated intracranial pressure does to the human eye. Various countermeasures have been studied. sleeping with the head elevated, using a posture device that raises the upper body angle during sleep, the period of most intracranial pressure accumulation, shows modest benefit in small studies. Lower body, negative pressure, the vacuum shorts device that draws fluid toward the legs, temporarily reduces intracranial pressure and may help. Pharmacological interventions that reduce cerebrospinal fluid production have been explored, though their long-term safety profiles in this context are unknown. None of these approaches has been proven to prevent SANS entirely, and none has been tested over the multi-year timescales relevant to deep space missions. The body does not stop there. The trillions of microorganisms living within you are also affected in ways we are only beginning to understand. You are not, by cell count, mostly human. The human body contains approximately 37 trillion human cells: neurons, hepatocytes, cardiomyocytes, epithelial cells, red blood cells, and also approximately 38 trillion microbial cells. Bacteria that live in every compartment of your gut, from stomach to colon, fungi on your skin, viruses integrated into your genome. This microbial community, the human microbiome, is not a passenger. It is a participant. The gut microbiome in particular performs functions that your human cells cannot perform alone. Fermenting dietary fiber into short-chain fatty acids that nourish the colon lining, synthesizing vitamins B12 and K2, training the developing immune system to distinguish self from non-self, and maintaining the integrity of the intestinal barrier that prevents bacterial translocation into the bloodstream. The microbiome is also exquisitely sensitive to perturbation. Change the diet, add stress, alter the sleep cycle, administer antibiotics, introduce a new pathogen, change the gravitational environment and the physical forces acting on gut motility. Any of these inputs can shift the composition of the microbial community, and a shifted community often means a compromised community, less diverse, less resilient, and less capable of performing its normal range of functions. Studies of ISS astronauts have documented significant microbiome changes during spaceflight. Certain bacterial families, including some associated with inflammatory signaling, increase in relative abundance. Others, including many lactobacillus and bifidobacterium species, associated with immune regulation and protection against pathogens, decline. The overall diversity of the community, the number of distinct species present, tends to fall. A less diverse gut microbiome is generally considered a less healthy gut microbiome. The analogy to ecology is apt. An ecosystem with fewer species is less stable, less capable of recovering from perturbation, and more vulnerable to takeover by opportunistic species. The immune consequences of this microbial shift are among the most clinically concerning aspects of spaceflight physiology. The immune system of long-duration spaceflight crew members shows consistent dysfunction across multiple measurement approaches. Natural killer cell activity, the surveillance function that destroys virally infected cells and tumor cells before they establish themselves, is reduced. Cytokine profiles are altered in patterns associated with chronic low-grade inflammation, and latent viruses begin to escape from immune control and reactivate. In a healthy person on Earth, Epstein-Barr virus sits silently in the lymphocytes, held in check by T-cell surveillance. Varicella zoster sleeps in dorsal root ganglia, suppressed by circulating immunity. During long-duration spaceflight, both of these viruses, and several others in the herpes virus family, reactivate and begin shedding in the saliva and urine of a significant fraction of astronauts. This is measurable. It has been measured. It represents a real, documented failure of immune surveillance in people who, by every other clinical measure, appear healthy. What this means for a deep space mission depends heavily on circumstances that are difficult to predict in advance. If the crew members are well selected for immune health and start with strong baseline immune function, the erosion during the mission may be tolerable. If one crew member is carrying a pathogen that their immune system has been controlling, and their immune surveillance weakens enough during the mission for that pathogen to establish active infection, the situation becomes more serious. And if an infection establishes itself in a crew member months into a Mars transit, with the nearest hospital not just hours away but a communications delay and an orbital mechanics problem away, the consequences of that infection cascading into something serious are significant. The psychological dimension of deep space travel is, by most accounts of the researchers who have spent careers studying isolated, confined environments, the variable with the most potential to derail a mission entirely. Not because astronauts are psychologically fragile. They are selected specifically for psychological resilience, emotional stability, and behavioral adaptability under stress. But because the environment of a deep space mission is so profoundly unlike any environment, human psychology evolved to handle that even the most psychologically robust individuals will face pressures that have no real analog in any life experience or training program. In 1970, the crew of Apollo 13 looked out the window of their spacecraft into the dark and knew their oxygen tank had exploded. They knew their primary mission was over. They knew they might not make it home. They were, in those first terrifying hours, more alone than any human beings had ever been. 321,000 kilometers from Earth, in a crippled spacecraft with no guarantee of survival. Jack Swigert, Fred Hayes, and Jim Lovell survived. They survived because of extraordinary technical ingenuity on both their part and that of the team on the ground. And crucially, they survived with the psychological support of constant, real-time communication with hundreds of people in Houston who were working the problem alongside them. Gene Kranz's team at Mission Control, famous for the declaration that failure was not an option, was available every second. Every calculation, every decision, every option could be discussed and checked in real time. A crew bound for Mars cannot have that. At the distance of Mars, a radio signal takes between 4 and 24 minutes each way. A crew facing an emergency sends a message and then waits. In a crippled spacecraft, in the middle of an unfolding crisis, for up to 48 minutes for any response. In a situation where seconds matter, there is no mission control. There is no expert on the other end of the radio who can help right now. The crew is on their own in a way that no crew in the history of human spaceflight has ever been. This is a genuine rupture from everything that has gone before in human spaceflight. Every mission, from Gagarin's 108-minute orbit to the year-long ISS expeditions of the modern era, has been conducted within a framework of rapid communication with Earth. Astronauts have always had mission controllers on voice loops, available within the speed of light. The psychological architecture of human spaceflight, the culture of flight rules, of constant monitoring, of decisions coordinated with the ground team, is built entirely around that real-time communication capability. Remove it, and the crew must become entirely self-sufficient, not just technically, but psychologically. They must make decisions without external validation. They must diagnose medical conditions without a physician on call. They must manage interpersonal conflicts without a mediator available by radio. They must maintain their own mental health without access to real-time psychological support. And they must do all of this while living in a volume perhaps comparable to the interior of a school bus, with the same 4 to 6 people for 2 to 3 years, with no possibility of going anywhere else. The research on isolated, confined environments tells a consistent story about what this does to groups of people. The Mars 500 experiment in Moscow simulated a 520-day Mars mission with a crew of 6 men confined in a habitat module complex. The data gathered from that experiment, including continuous activity monitoring, detailed behavioral observation, and physiological measurements, revealed a profile of psychological deterioration that matched what had been observed in Antarctic overwinter crews, submarine crews, and polar expedition teams. Participation started strongly. The crew was engaged, motivated, productive in the early weeks. Then the middle phase arrived, the long, undifferentiated middle where the novelty had worn off. The end was not yet visible, and the mission had settled into a grinding daily routine. Activity levels fell. Sleep increased dramatically in some crew members, with one participant averaging over 12 hours of sleep per day during certain phases. Interpersonal dynamics became more complicated. Social withdrawal increased. Participation in group activities declined. And critically, the Mars 500 crew knew they were in Moscow. They knew there was a door. They knew the experiment could end. They knew medical care was available outside the habitat. A real Mars crew would have none of these psychological safety nets, and the absence of those nets is not a trivial difference. The knowledge that emergency escape is possible fundamentally changes the psychological experience of confinement, even if that escape is never actually used. Nick Kanas, the psychiatrist who has spent decades researching astronaut psychology, has identified several specific risk factors that are present in deep space missions in uniquely acute form. The communication delay breaks the normal feedback loop of social interaction, the timely exchange of information, the real-time calibration of relationships, in a way that communication with Earth can no longer provide. The crew becomes, psychologically, a closed system. Their social universe consists of the few people in the spacecraft. The quality of those relationships and the health of the group dynamics within that small community becomes the entire social world. In a closed social system under prolonged stress, small conflicts become large ones. Personality differences that are manageable in a large organization become points of friction in a confined group. The things that differ between people, their risk tolerance, their communication styles, their responses to authority, their hygiene habits, their sleep schedules, all of these become subjects of daily negotiation in a way they never are in normal social environments, where the option to simply avoid the irritating person always exists. On a deep space spacecraft, you cannot avoid anyone. The person whose chewing you find irritating is sleeping 3 meters away from you. The person whose risk assessments you disagree with is also the person you are relying on to operate critical systems. The person whose emotional needs feel overwhelming at the end of a 16-hour workday is also the person whose well-being is your responsibility. There is no leaving. There is no getting space. There is only the spacecraft and the mission and each other. On August 4th, 1972, something happened between the Apollo 16 and Apollo 17 missions. Something that barely made the news at the time, that most people have never heard of, and that radiation physicists still talk about as one of the most significant near misses in the history of human spaceflight. The sun erupted, a massive solar flare, accompanied by a coronal mass ejection. An enormous cloud of magnetized plasma ejected from the solar surface with the energy of billions of nuclear weapons, swept through the inner solar system at several million kilometers per hour. The cloud of energetic protons that preceded it, accelerated by the shock wave of the flare, traveled faster still. By extraordinary coincidence, the timing fell between missions. Apollo 16 was already back on Earth. Apollo 17 was still being prepared for launch. The narrow window between those two missions happened to coincide almost exactly with the arrival of one of the most energetic solar particle events recorded in the modern era of space weather observation. Engineers calculated afterward, based on the measured particle flux and energy spectrum of that event, that a crew inside a standard Apollo command module in deep space during that storm would have received a whole-body radiation dose exceeding 10,000 millisieverts in the span of a few hours. The NASA lifetime dose limit for astronauts is currently around 600 millisieverts. the crew would have absorbed 17 times that limit in an afternoon. At those dose levels, acute radiation syndrome is not a risk. It is a certainty. The crew would have experienced nausea and vomiting within hours. Their bone marrow, which produces all the blood cells that sustain life, would have been destroyed within days. Without access to a transplant facility, without intensive medical support, they would have died within weeks. This nearly happened. A 4-day shift in the launch schedule, a 4-day delay in the previous mission's return, and 12 people would have died in deep space in one of the most dramatic disasters in the history of exploration. Solar particle events like the 1972 storm are not rare flukes. They are a predictable feature of the solar activity cycle, most common near solar maximum, but possible at any phase. They cannot be reliably predicted more than minutes to hours in advance. A crew in deep space has, at most, perhaps an hour of warning before the most energetic protons arrive, time enough to shelter, if the shelter is immediately accessible and the crew acts without hesitation. Time not enough if a crew member is on a spacewalk, or if the shelter is blocked, or if the monitoring systems have any failure. The physics of solar particle event shielding is, like the physics of galactic cosmic ray shielding, constrained by the same fundamental problem. Adequate shielding requires mass, and mass is prohibitively expensive to launch. The radiation shelter on a planned deep space spacecraft, a compact compartment surrounded by water tanks and food stores and other hydrogen-rich materials, can reduce the dose from a large SPE by a factor of 2 or 3. In a relatively modest event, that reduction may be enough to avoid acute radiation syndrome. In a 1972 scale event, no practically launchable shielding would be adequate. The honest position of space radiation safety experts is that a deep space crew will be exposed to one or more significant solar particle events during a multi-year mission, that the spacecraft shielding will not provide complete protection, and that the crew will have to accept some probability of receiving a dose high enough to cause acute symptoms. How high that probability is, and how severe those symptoms might be, depends on the specific mission timeline, the spacecraft design, the solar cycle phase at the time of the mission, and random chance. Life support presents its own set of challenges that accumulate alongside the biological ones. The average adult human consumes approximately 550 liters of oxygen per day, exhales around 400 liters of carbon dioxide, and requires about 2 liters of water. Multiply by 6 crew members, multiply by 3 years. The total oxygen requirement approaches 1 billion liters at standard temperature and pressure. The total carbon dioxide production approaches 700 million liters. The total water requirement exceeds 13,000 liters per person. None of this can be resupplied. The mission carries everything it needs, or it generates what it needs from what it carries, or it fails. The ISS has demonstrated that a closed-loop life support system capable of sustaining human life for years is achievable. Electrolysis produces oxygen from water. The Sabatier reaction recovers some of that water from exhaled carbon dioxide. Chemical scrubbers absorb carbon dioxide when regenerative systems are insufficient. Water recovery technology processes urine, perspiration, and exhaled moisture into potable water of exceptional purity. These systems are complex. They require maintenance. They have failure modes. They have been refined over decades of operation on the ISS, and they still require regular maintenance and occasional emergency intervention. On a deep space spacecraft, the same systems must operate for years, with no possibility of resupply from Earth, and with repair capability limited to whatever spare parts and crew expertise the mission carries. If the oxygen generation system fails, the crew has emergency oxygen reserves, but only for a limited time. If the water recovery system degrades below a certain efficiency, the water reserves begin depleting in a direction that cannot be reversed without intervention. If the carbon dioxide scrubbing system has a failure that the crew cannot repair, CO2 begins accumulating in the cabin air. Carbon dioxide management is subtler and more insidious than oxygen management. While oxygen deprivation causes obvious dramatic symptoms quickly, the body and brain register hypoxia rapidly and loudly. Elevated CO2 causes effects that can be difficult to notice because they mimic the normal cognitive state. Mild hypercapnia causes headaches that feel like ordinary stress headaches. It causes fatigue that feels like ordinary mission fatigue. It causes slight cognitive slowing that feels like ordinary end-of-workday mental tiredness. A crew member experiencing mild CO2 toxicity might feel vaguely unwell without identifying the cause. If the CO2 level has been gradually rising for days, the crew may have been adapting to a slowly worsening baseline without recognizing that their cognitive function has been impaired. Food is the life support factor that shapes daily psychological experience more than any other. Nutrition can be engineered, calories can be calculated and provided with precision. But food is not merely fuel. The preparation and sharing of meals is one of the oldest and most universal bonding behaviors in the human species. It signals care, community, safety, and celebration. Anthropologists studying hunter-gatherer societies have found that the amount of time spent in communal food preparation and eating is correlated with social cohesion and group function. For our species, eating together is not optional. It is structural. The freeze-dried and thermostabilized food available on current spacecraft is nutritionally adequate but sensory impoverished. The fluid shift that affects astronauts throughout their missions creates a chronic congestion of the sinuses that permanently dulls the sense of smell. And smell is responsible for the majority of what we consciously experience as taste. A strawberry that would taste intensely sweet and fragrant on Earth tastes like sweetened cardboard in microgravity, not because the food has changed, but because the astronaut's sensory apparatus has been degraded by the physiological environment. Eating becomes a functional act rather than a pleasurable one. Over months, this has measurable psychological consequences. In March 2015, astronaut Scott Kelly rode to the International Space Station for what would become a year-long mission that his twin brother Mark Kelly, also a retired astronaut, would experience vicariously from the ground as the other half of the most scientifically valuable natural experiment in the history of human spaceflight research. For 12 months, both brothers underwent identical batteries of tests at identical intervals. Blood samples processed for genomic and epigenomic analysis. Stool samples for gut microbiome characterization using advanced metagenomic sequencing. Cognitive assessments using standardized neuropsychological testing batteries. Continuous sleep monitoring. Echocardiographic assessment of cardiac structure and function. Carotid artery imaging for wall thickness measurement. Telomere length analysis from white blood cells. Everything that modern biomedicine could measure was measured in both of them simultaneously, so that the changes in Scott could be attributed to spaceflight rather than to aging, seasonal variation, or any other confounding factor, because his genetically identical twin was right there on Earth providing the comparison. The results of the twin study, published in 2019 across a series of papers in the journal Science, were comprehensive and in places surprising. Scott Kelly's telomeres, the repetitive DNA sequences that cap the ends of chromosomes and whose length is used as a marker of cellular aging, had lengthened during the mission. Longer telomeres are associated with younger, healthier cells. This finding was initially presented as paradoxically positive news for spaceflight biology, but the longer-term picture was darker. After Scott returned to Earth, his telomere length declined rapidly and within months had fallen below his pre-flight baseline, and below Mark's telomere length, which had been gradually declining normally with age on Earth. The temporary lengthening in space appeared to have been followed by an accelerated shortening on return. The net effect over the full observation period was that Scott Kelly's cells showed signs of accelerated aging relative to his identical twin who had stayed on the ground. More than 900 genes showed altered expression patterns in Scott's white blood cells during and after the mission, compared to Mark's. The activated pathways included DNA damage response, the body's cellular fire brigade working overtime on the damage accumulated from months of radiation exposure, as well as immune activation pathways and genes associated with hypoxia response and bone metabolism. Some of these expression changes persisted for 6 months after return, suggesting that a year in space had caused epigenetic remodeling. Changes not to the DNA sequence itself, but to the pattern of chemical modifications on the DNA that determine which genes are read and which are silenced. That did not fully revert when the spaceflight environment was removed. His gut microbiome showed dramatic compositional changes during the mission, with partial but incomplete restoration of the pre-flight community afterward. His cognitive performance on standardized tests, particularly tests of speed and accuracy, showed subtle but measurable declines that were still present at the 6-month post-return assessment. His carotid artery wall thickness, measured by ultrasound, had increased during and after the mission, a pre-atherosclerotic change that his brother on Earth had not experienced over the same period. None of these findings, considered individually, add up to a catastrophic health outcome. Scott Kelly is not permanently disabled. He has not developed cancer. He functions normally by any everyday standard. The scientists who conducted the twin study were clear and measured in their conclusions. The changes they documented were biologically significant, but did not suggest that long-duration spaceflight is incompatible with subsequent health. But that is not the question the twin study was really asking. The question it was asking is, what does space do to the human body that Earth does not? And the answer it returned was, more than anyone had previously measured in more body systems, more persistently, and with a degree of individual variation that makes predicting outcomes for any specific astronaut very difficult. And the twin study examined 12 months in low Earth orbit within partial magnetospheric protection, with the best current countermeasures in place. A Mars mission is 2 to 3 years in deep space, with radiation levels 3 to 5 times higher, with no magnetospheric protection, and with countermeasures that for many of the identified problems are still incomplete or unproven. Stand on a beach on a clear night and look up. The moon is there, sometimes, 250,000 miles away. In terms of human spaceflight history, we have been there. 12 people walked on its surface between 1969 and 1972. The farthest any human beings have ever traveled from Earth is approximately 400,171 kilometers, set by the Apollo 13 crew as they looped around the far side of the moon on their aborted mission. Mars, at its closest approach to Earth, is roughly 200 times farther than the moon. The moons of Jupiter, at their closest, are more than 2,500 times farther. Saturn's orbit is nearly 4,000 times farther. The nearest star beyond the Sun is approximately 100 million times farther than the Moon. We have not been to Mars. We have never been farther from Earth than the Moon. Every astronaut who has ever flown spent their entire spaceflight career within the partial protection of Earth's magnetosphere, within reliable real-time communications distance of Earth, within a feasible emergency return window. The data we have on human spaceflight physiology comes entirely from this protected close-in regime. The moment a spacecraft crosses the magnetopause and enters true interplanetary space, it enters a domain where no human biological data exists. We are extrapolating from ISS studies and animal experiments and analog environments and physical models and hoping that the extrapolations hold. What history suggests is that the extrapolations will hold partially. Some of what we've predicted from ISS data will be roughly correct for deep space. Some problems will be worse than expected. And some problems, like SANS was before we noticed it, like the 1972 solar storm was before it almost killed a crew, will be things we haven't predicted at all. The people who are trying to solve all of this are not deterred by this uncertainty. They are motivated by it. There are radiation biologists who have structured their entire research careers around the question of how heavy ion radiation damages neural tissue and whether that damage can be pharmacologically mitigated. There are mechanical engineers designing rotating habitats and running simulations of how partial artificial gravity would affect the bone and cardiovascular physiology problems that exercise alone cannot solve. There are space medicine physicians developing autonomous medical capability protocols, detailed AI-assisted decision trees for diagnosing and treating every medical condition a crew might plausibly face during a multi-year deep space mission without any real-time physician consultation available. There are food scientists formulating hydroponic crop systems for spacecraft that could provide fresh vegetables in volumes large enough to meaningfully supplement the freeze-dried food supply, improving both nutrition and psychological quality of life. There are psychologists developing pre-mission crew composition algorithms, selecting team configurations that minimize interpersonal conflict risk based on personality profiles, communication styles, and conflict resolution patterns. And in mission psychological support protocols designed to maintain crew mental health across multi-year durations. None of them will tell you the problems are solved. All of them will tell you that every specific problem has a specific solution, and that the solution is a matter of doing the work and finding the answer, not of accepting that the barrier is permanent. Dr. Jessica Meir, who has spent months aboard the ISS, and who has returned to Earth and rebuilt her physiology through the weeks of recovery that long-duration spaceflight requires, has described what floating in the cupola and watching the Earth rotate below, watching an entire continent drift past in the time it takes to eat lunch, does to the relationship between a person and their species. It makes the abstract concrete in a way that is very difficult to convey. The human footprint on the planet is visible. The atmosphere is visible as a thin, precious blue line above the horizon. The darkness beyond it is visible, absolute and deep and permanent. You understand, in a way that you cannot fully understand from the ground, how rare and fragile and unlikely this particular arrangement of matter is. The people who have been there come back changed, not just physiologically in the ways we have been discussing, but in the more interior sense that all significant experiences change a person. They come back with a perspective that is genuinely different from the one they left with. And some of them, having absorbed that perspective, go back, voluntarily, knowing what they know about what it does to the body, knowing the radiation numbers, knowing the bone density curves, knowing the intracranial pressure findings. They go back anyway. The real reason no one can survive deep space indefinitely, as things currently stand, is not a single failure mode. It is not just the radiation, or just the bone loss, or just the vision damage, or just the isolation, or just the immune dysfunction. It is the accumulation of all of them, operating simultaneously every day without pause for the entire duration of the mission. Space does not take days off. Every day you are out there, the galactic cosmic rays are threading through your neural tissue. Every day the absence of gravity is demanding calcium from your hip bones. Every day, the elevated intracranial pressure is pressing against your optic nerves. Every day the confined artificial environment is working on your microbiome and your immune system. Every day the psychological weight of isolation and responsibility and distance accumulates. The human body is not fragile. 3.5 billion years of evolution produce something capable of surviving ice ages and famines and tropical diseases and high-altitude environments and the deepest ocean trenches. It produced something that can spend a year in orbit and walk away. But it did not produce something designed for indefinite operation in the void beyond Earth. It produced something designed for this planet, and this planet only. Whether that is the end of the story or the beginning of the next chapter is the question that drives everyone who works in human spaceflight. The answer is not yet known, but the answer is being actively sought in laboratories and simulation chambers and research institutes and analog environments across the world by people who believe that the problem is not impossibility, but incompletion. That we are not at the edge of what is possible. We are at the edge of what has been done. There is a moment in every extended spacewalk that astronauts describe in almost identical terms, regardless of their nationality, their background, or the specifics of their mission. It comes when they stop moving, stop working, release the handrail, and simply float. They look at the Earth below them, that impossible blue, the clouds spiraling in slow white curls, the dark of the deep ocean and the warm texture of the continents. They look at the stars above, unwinking, unfiltered by any atmosphere, brilliant and still in a way you cannot experience from the ground. They look at the black between the stars, the deep black, the kind of black you don't get on Earth even in the most remote desert on the clearest night. The black that has no bottom and no edge and no end. And for a moment, they feel very small. And for a moment, they feel very large. And for a moment, those two things are not contradictory at all. The void is hostile. We have spent this entire film proving that in detail. It will damage your eyes and weaken your bones and blunt your immune response and alter your DNA and quietly remodel your cardiovascular system and test your mind to limits that no amount of training can fully prepare you for. It was not built for you. It does not want you there. And if you go far enough and stay long enough without better solutions than the ones we currently have, it will kill you. But people go anyway. They have gone since Gagarin, and they will go further. And the reasons they go are not fully reducible to mission objectives, or scientific curiosity, or national competition, or the logic of exploration. There is something in the act of leaving Earth that speaks to something very old in the human animal. Something that preceded the telescope, and the rocket, and the space station by millions of years. Something that cannot look at a horizon without needing eventually to cross it. The problems are real. The risks are real. The costs to the body, to the mind, to the years of life that radiation exposure and physiological stress extract from those who go are real. And still, people go. That might be the most extraordinary thing about the entire enterprise of human spaceflight. Not the engineering, not the science, not even the destinations. The fact that we know the cost, and we go anyway. That might be the most human thing of all. You are not, by cell count, mostly human. The human body contains approximately 37 trillion human cells. It also contains roughly 38 trillion microbial cells, bacteria, archaea, fungi, and viruses that have lived alongside the human lineage for millions of years. They have never, in the entire history of their co-evolution with us, experienced zero gravity. They did not evolve for it, and when you take them there, they change, sometimes in ways we understand, often in ways we don't. The gut microbiome is perhaps the most critical component of this microbial community from a health perspective. It is involved in digesting food, particularly the complex plant fibers that human enzymes cannot break down alone. It synthesizes vitamins the body cannot produce itself. It trains the immune system during development to distinguish self from non-self. It maintains the physical integrity of the intestinal lining that separates the contents of the gut from the bloodstream. It produces signaling molecules that communicate with the brain via the vagus nerve, influencing mood, stress response, and anxiety levels. The gut microbiome is not a passenger in your body. It is a participant in your physiology. Studies of astronauts on the International Space Station have documented consistent and significant changes in gut microbiome composition during spaceflight. Species-level analyses using advanced sequencing technology showed that the community shifts in characteristic ways. Certain potentially inflammatory bacterial families increase. The beneficial organisms associated with short-chain fatty acid production, immune regulation, and intestinal barrier maintenance decline in relative abundance. The overall diversity of the community, the raw number of distinct species present, falls. A less diverse microbiome is a less stable and less functional microbiome, in the same way that an ecosystem with fewer species is less resilient and more vulnerable to disturbance. The immune consequences of this microbial disruption add to the direct immunological effects of radiation and stress. Long-duration astronauts show consistent immune dysfunction across multiple measurement approaches. Natural killer cell activity is reduced. The cytokine balance, the system of chemical messengers that coordinates immune responses, shifts toward a chronic inflammatory pattern, and latent viruses escape from immune control and begin shedding. In a healthy person on Earth, Epstein-Barr virus sits silently in memory B cells, held in check by cytotoxic T lymphocytes that patrol the body looking for signs of reactivation. Varicella zoster sleeps in the sensory nerve ganglia, suppressed by circulating antibody and T-cell surveillance. During long-duration spaceflight, both of these viruses reactivate and begin appearing in saliva and urine samples. The same has been documented for cytomegalovirus and herpes simplex. These are not hypothetical risks inferred from animal studies. They are documented observations from actual astronauts on actual missions. The immune system that normally holds these viruses in check is failing to do so. For a multi-year deep space mission, this pattern of immune erosion raises a specific set of operational concerns. If immune surveillance is progressively weakening over the course of the mission, conditions that are normally contained by immune control may begin to manifest clinically. A crew member who has been managing a low-level Epstein-Barr reactivation silently may develop symptomatic mononucleosis-like illness partway through the mission. A crew member carrying latent tuberculosis, which is present in roughly a quarter of the world's population in dormant form, might develop active TB if the suppression of the immune response reaches a threshold that allows the bacteria to begin replicating. The medical management of these scenarios on a spacecraft is severely limited. The formulary of medications that can be practically carried is constrained by weight, volume, and shelf life. Some antibiotics lose significant potency before a 3-year mission is complete. Antiviral drugs for herpes virus reactivation are available and relatively stable, but managing a seriously ill crew member with limited nursing support and no physician on call on a spacecraft hundreds of millions of kilometers from Earth is a different challenge from managing the same illness in a hospital. Mission planners working on deep space human health have developed the concept of crew medical officer training, giving designated crew members substantially more medical education than has historically been required for spaceflight. Some proposals call for CMO-level crew members to have training roughly equivalent to a physician's assistant or family nurse practitioner, with focused additional training in the specific conditions most likely to arise during the mission. Telemedicine support from Earth, delayed by the communication lag, can provide guidance, but the crew member performing the procedure or making the treatment decision is doing so in real time, with information from Earth arriving after the fact. The psychological dimension of performing medical care on a crewmate in deep space has not been fully studied. We know from terrestrial research in isolated environments, expedition medicine in polar regions, medical care on maritime vessels at sea, that even trained medical personnel experience significant psychological stress when providing care to colleagues in remote settings without backup support. The additional factors of communication delay, spacecraft confinement, and the impossibility of medical evacuation compound that stress substantially. Now, let's talk about what gravity actually gives you. Beyond just holding you to the floor, artificial gravity, using the centrifugal effect of rotation to simulate gravitational loading, is the conceptual solution to the microgravity problem that engineers and mission planners have been thinking about since before any human ever went to space. Wernher von Braun, who became the chief architect of NASA's moon program, was writing detailed technical proposals for rotating space stations in the early 1950s. Stanley Kubrick's 2001, A Space Odyssey, depicted a rotating space station with technically accurate artificial gravity in 1968, a year before the moon landing. The concept is as old as the idea of long-duration spaceflight itself. The physics is simple. A rotating structure generates centripetal acceleration, the apparent outward force that keeps you pressed to the inner wall of a rotating cylinder. Make the rotation fast enough and the radius large enough, and the centripetal acceleration equals the gravitational acceleration of Earth. The floor pushes up on your feet. Your skeleton loads normally. Your cardiovascular system has to pump against a gradient. Your inner ear senses a consistent up-down orientation. All of the problems of microgravity physiology, in principle, go away. The engineering difficulty is that humans are sensitive to rotation. The vestibular system in the inner ear detects rotation and can cause motion sickness at rotation rates above approximately 2 to 4 revolutions per minute, depending on the individual and the adaptation time. To generate Earth-equivalent gravity at only 2 revolutions per minute, you need a rotation radius of approximately 224 meters, a structure nearly half a kilometer in diameter. That is far beyond any spacecraft architecture currently imaginable for the near future. At smaller radii, faster rotation is required to generate the same gravitational acceleration. A radius of 20 meters requires rotation at about 6.7 revolutions per minute to produce Earth gravity. Most people cannot tolerate that rotation rate, at least not without significant acclimatization. And a more fundamental problem arises, the Coriolis effect. In a rotating reference frame, moving your head up or down when the rotation axis is horizontal generates an apparent sideways force that the vestibular system perceives as a conflicting signal. This creates disorientation and nausea that limits the usable rotation rate, regardless of acclimatization. Some researchers believe that acclimatization can push the tolerable rotation rate substantially higher, that people can adapt to 4, 5, or more revolutions per minute given gradual exposure. If so, a much more compact artificial gravity system becomes possible. A habitat module 10 to 15 meters in radius, rotating at 4 to 5 revolutions per minute, could generate roughly half of Earth's surface gravity, enough perhaps to prevent the worst physiological consequences of microgravity, without requiring a structure larger than anything we've ever launched. The question of how much artificial gravity is enough has not been answered, because it has never been tested at any duration. We don't know whether 1/3 of Earth's gravity, Mars-level gravity, is sufficient to prevent bone loss. We don't know whether half of Earth's gravity prevents cardiovascular deconditioning. We don't know whether any partial gravity is sufficient to reverse the fluid shift and prevent SANS, or whether something close to full Earth gravity is required for that. The animal research on partial gravity is sparse, conflicting, and done at short durations. The human research essentially doesn't exist at durations relevant to a Mars mission. This knowledge gap is one of the most significant gaps in human spaceflight medicine. It is also one of the most tractable to fill in principle. A rotating habitat in Earth orbit inhabited by research crews for periods of months, could generate the data needed to answer the partial gravity question definitively. The technical challenge is modest compared to a Mars mission. The scientific return would be enormous. This type of research station has been proposed multiple times. It has never been funded and built. The reason is always the same: money and priorities. The cost of building and operating a rotating research station in Earth orbit, while not absurd relative to the overall cost of human spaceflight, is real, and it is funding competition for hardware and operations that the existing space station programs already consume. Until there is a firm commitment to a deep space human mission that makes the artificial gravity question urgent rather than academic, the rotating research station remains a proposal. Meanwhile, the people planning deep space missions work with what they know: that microgravity is physiologically damaging over the time scales relevant to a Mars mission, that exercise countermeasures are helpful but insufficient, and that the mission will proceed with a population of crew members whose physiology is knowingly being compromised in ways that may not be fully reversible. Sleep in space is its own underappreciated challenge. The human circadian rhythm, the roughly 24-hour biological clock that regulates the sleep-wake cycle, hormone secretion, body temperature, digestion, and dozens of other physiological processes, is synchronized primarily to light. The photoreceptors in the retina detect the blue wavelength light of daylight and use it to set the master clock in the suprachiasmatic nucleus of the hypothalamus. As daylight fades and blue light decreases in the evening, the pineal gland releases melatonin, which signals all of the body's clocks to begin the sleep preparation sequence. On the International Space Station, the orbital period is approximately 90 minutes. The station experiences a full sunrise and sunset every 90 minutes. 16 sunrises and 16 sunsets per 24-hour day. The circadian system, which evolved for a 24-hour day with one long period of darkness, receives completely contradictory light signals from the environment. It cannot entrain to a 90-minute cycle. Astronauts on the ISS maintain an artificial 24-hour light schedule using the station's internal lighting systems and by covering the windows, but the quality of circadian entrainment is poor. Astronaut sleep studies consistently document shortened sleep duration, reduced sleep quality, more frequent waking, and misalignment between the biological clock and the scheduled sleep time. On a deep space transit spacecraft, the light environment can be fully controlled. There are no 90-minute orbital sunrises to disrupt the schedule. In this respect, deep space transit may actually be better for sleep regulation than ISS. But the psychological factors that degrade sleep on long-duration ISS missions, the stress, the elevated CO2, the noise of life support equipment, the confined sleeping quarters, the inability to fully mentally disengage from the mission's constant demands, will be present on a deep space spacecraft as well. Chronic sleep deprivation, even mild sleep deprivation, has cognitive consequences that accumulate over time. Studies of sustained inadequate sleep show that people develop stable cognitive deficits, impaired attention, slower reaction time, reduced working memory capacity, impaired emotional regulation, that they progressively stop noticing because the impaired state becomes their new baseline. They are less capable than they were before the sleep deprivation began, but they feel normal because they have adapted to the new normal. They don't know they're impaired. For a crew operating a complex spacecraft while making life or death decisions in real time, this kind of invisible cognitive degradation is a genuine safety concern. The checklist culture of human spaceflight, the reliance on detailed procedures and protocols rather than improvisation, the multiple layers of verification before any consequential action, is partly designed to compensate for the cognitive impairments that stress and fatigue inevitably impose on human operators. But checklists and procedures protect against the errors that operators know to check for. They don't protect against the novel situations, the unexpected emergencies, the off-nominal scenarios that the procedures don't cover. Where clear, rapid, creative thinking is required, the distance between where we are and where we need to be to send humans safely and sustainably into deep space is very real. It is measured not in the single barrier of one intractable problem, but in the accumulation of dozens of problems, each of which has partial solutions that are individually insufficient and collectively imperfect. Radiation, microgravity physiology, SANS, immune dysfunction, microbiome disruption, psychological isolation, sleep quality, autonomous medical care, food quality and variety, water and oxygen system reliability, communication delay. Each of these is a chapter in a textbook that is still being written, and yet the trajectory of progress is clear. Each year, the understanding of each problem deepens. Each year, the countermeasure options improve. Each year, the technologies that might solve the hardest problems become more mature. Pharmacological radiation protection is getting closer. Artificial gravity concepts are getting better defined. The microbiome science is advancing rapidly. The psychological support systems being developed for long-duration missions are more sophisticated than anything that existed a decade ago. The scientists and engineers working on these problems are not discouraged by the scale of what remains to be done. They are precisely the kind of people who respond to large, unsolved problems with engagement rather than retreat. They have chosen careers defined by the hardest questions in human spaceflight medicine, and the hardest questions are what sustains them. There are radiation biologists at the NASA Space Radiation Laboratory at Brookhaven National Laboratory who have spent entire careers understanding what heavy ions do to biological tissue, testing compound after compound in the search for neural protection, accumulating the deep mechanistic knowledge that a pharmacological solution will eventually require. There are aerospace engineers at multiple space agencies designing habitat concepts that include rotation for artificial gravity, running structural and system simulations, refining the architecture towards something that could actually be built and launched. There are human factors, researchers developing the new framework for crew autonomy in deep space, the protocols, the decision-making structures, the crew organization models that work when Earth is 45 minutes away from the nearest possible response. There are food technologists developing plant growth systems optimized for spacecraft volume and energy constraints, crop varieties selected for nutritional density and palatability in the sensory environment of long-duration spaceflight. There are telemedicine specialists building the diagnostic and treatment decision systems that a crew medical officer in deep space will use to navigate medical situations without real-time physician consultation. All of these threads of research and development are converging on the same eventual capability: the ability to send humans into deep space for years at a time and have them return healthy or healthy enough, in a way that honors the extraordinary commitment they have made and the extraordinary risk they have accepted. The people who will go on the first crewed Mars mission, whoever they are, whenever that mission flies, will go knowing the full scope of what the journey will do to their bodies and their minds. They will know the radiation numbers. They will have seen the bone density curves. They will have read the SANS literature. They will know that their telomeres may be shortened, and their cognitive performance may be slightly diminished, and their eyes may have changed in ways that don't fully reverse. They will know that they will come back physiologically older than the calendar would suggest. And they will go anyway, not because they don't understand the costs, but because they have decided, each of them individually and all of them together as a crew, that the purpose of the journey is worth the cost. That the thing to be learned, the thing to be seen, the thing to be proven, the thing to be accomplished by standing on the surface of another planet and looking back at a point of blue light in the sky, and knowing that everything human history has ever produced happened on that point. That thing is worth what it costs to get there. That calculation made by real people with full information about the consequences is the most remarkable thing about human spaceflight. Not the rockets, not the spacecraft, not the scientific return, the willingness of human beings, knowing what they know, to go anyway. There is a moment in every extended spacewalk that astronauts describe in almost identical terms, regardless of their background or nationality or mission. It comes when they stop working and simply float. They look at the Earth below, that impossible blue, the clouds in slow white spirals, the deep dark of ocean and the warm brown of desert. They look at the stars above, burning steadily in the absolute black, unobscured by atmosphere, more real and more present than they ever appear from the ground. They look at the black between the stars, the deep black, the permanent black, the black that has no bottom. And for a moment, they feel very small. And for a moment, they feel very large. And for a moment, those two things are not opposites, but the same thing seen from different angles. The void is hostile. We have spent the last 2 hours proving that in exacting detail. It will damage your eyes and hollow your bones, and quiet your immune system, and tangle your DNA, and reshape your cardiovascular system, and press on your mind until the edges bend. It was not made for you. It was not made for anything alive. And if you go far enough into it and stay long enough with the tools we currently have, it will kill you. But people go anyway. They have gone since Gagarin, and they will go further. And the reasons they go exceed the logic of any mission objective or scientific return. There is something in the act of leaving the planet that speaks to something older than language, older than tools, older than fire. Something in the human animal that cannot see the dark without needing, eventually, to enter it. The problems are real, the risks are real, the costs are real, and still, people go. That willingness, clear-eyed, fully informed, absolutely irreducible, might be the most extraordinary thing about the entire enterprise. And it might be the most human thing of all. Consider for a moment what it means to be completely alone. Not the ordinary alone of a quiet evening or a solo hike. Not the alone of a retreatant in a monastery or a sailor on a passage. Something categorically different. An alone defined not just by absence of others, but by the impossibility of others. An alone in which every person you have ever known is on a point of light that your instruments can barely resolve at this distance. And the communications channel connecting you to that point introduces delays measured in tens of minutes, and the option of returning to that point is unavailable to you for the next 18 months. The psychological experience of deep space isolation has no precise terrestrial analog. The Antarctic winter-over cruise come closest perhaps. 8 to 12 people sealed into a station at the South Pole, unable to leave for the duration of the polar winter because the conditions outside are lethal and no aircraft can reach them. They are isolated, confined, cold, in perpetual darkness for months, with limited communication and no emergency evacuation possible. The psychological strain on these crews has been studied extensively. The findings are relevant to deep space mission planning, even though the analogy is imperfect. Antarctic overwinter crews consistently show a pattern of psychological change over the course of their isolation. In the early weeks, morale is high. The novelty of the extreme environment generates genuine engagement. People are performing at high levels, motivated by the challenge, bonded by the shared adventure of being somewhere very few people have ever been. Then, in the middle months, the novelty collapses. The darkness has been dark for a long time now. The cold is no longer interesting. The same faces appear at every meal, every briefing, every equipment check. The routines have become routines in the most grinding sense of the word. And the people, who were chosen for psychological stability and resilience, begin to show the effects of sustained confinement. Sleep disturbances become common. Emotional regulation becomes harder. Minor irritations that were easily brushed off in the early weeks have accumulated into genuine grievances. People become protective of whatever small private spaces and private routines they have. Motivation for non-essential tasks falls. The group dynamic shifts from collegial to careful. People managing their interactions more deliberately, avoiding certain conversations, being strategic rather than spontaneous. This is with 12 people in a structure that, while cramped, is substantially larger than a spacecraft. With regular communication to the outside world, albeit with some delay. With the concrete knowledge that winter will end, the planes will come and the outside world will return. With no mission-critical tasks that require split-second decision-making under extreme pressure, a deep space crew will have 4 to 6 people, not 12. They will have a spacecraft volume significantly smaller than an Antarctic station. They will have communication delays of up to 45 minutes each way. They will have no certain endpoint that their bodies know is coming and that their psychology can orient toward. They will have mission-critical tasks throughout the entire mission that cannot wait, cannot be deferred, and cannot be done incorrectly. The psychiatric community working on human spaceflight has developed a reasonably detailed picture of the psychological competencies that the most resilient isolated crew members share. They tend to have what psychologists call high trait openness, genuine intellectual curiosity and flexibility that sustains engagement with novel situations and problems over time. They tend to have stable, secure attachment styles. They can maintain emotional equilibrium without constant external validation. They tend to have strong executive function, the capacity to regulate emotion, manage competing priorities, and maintain behavioral consistency under sustained stress. They tend to be genuinely comfortable with uncertainty, not just tolerant of it, but capable of functioning well in situations where the outcome is unknown and the correct action is unclear. Selection processes for deep space missions will attempt to identify these traits. Pre-mission training programs will attempt to develop and reinforce them. Mission support protocols will attempt to maintain them across the duration of the mission. But even the most sophisticated selection and training and support process cannot guarantee that a given crew will navigate 2 or 3 years of deep space isolation without psychological casualties. The specific scenario that mission planners work hardest to plan around, and that some argue cannot be fully planned around, is a severe interpersonal conflict within the crew. Not ordinary friction, not the low-level irritation that any small group living in close quarters accumulates. A real, destabilizing conflict that impairs the function of 2 crew members and, by extension, the function of the entire crew. In a 6-person crew, a serious conflict between 2 members immediately affects the other 4, who must navigate their own relationships with both parties, manage the operational impact of the conflict on mission tasks, and maintain their own psychological stability while doing so. There is no mediator available. There is no authority figure who can separate the parties or impose a resolution. Mission Control can advise with a 45-minute round-trip communications delay, but cannot intervene. The crew has to solve its own problems in real time in a confined space with no exit. This is not a hypothetical scenario invented to frighten mission planners. It has happened in isolation experiments. It has happened in analog environments. Serious interpersonal conflicts that degraded the function of isolated groups have been documented in Antarctic crews, in submarine deployments, in underwater habitat experiments, and in the Mars 500 simulation. The frequency is low, the severity varies, but it is not zero, and it will not be zero in deep space. The countermeasures are primarily procedural and psychological. Pre-mission conflict resolution training teaches crew members how to manage disagreements constructively before the conflict escalates. Mission support protocols include scheduled psychological check-ins and access to confidential communication channels where crew members can discuss concerns without the entire crew hearing. Leadership selection and training focuses on building the specific skill set for managing a small group under chronic stress. Skills that are different from the technical expertise that traditionally defined the astronaut selection criteria. Some researchers have proposed pharmacological support for the psychological demands of deep space. Not antidepressants or antipsychotics, but targeted interventions that could help regulate anxiety, support sleep quality, or blunt the acute stress response during high-pressure moments. The ethical territory here is complex, pharmacologically altering the emotional and cognitive state of crew members who are also operating safety-critical systems, at a distance where any adverse reaction cannot be medically managed by anyone but themselves, requires very careful judgment about benefits and risks that the research base does not yet fully support. What is clear from the accumulated research is that psychological health in deep space is not a soft problem, not a quality of life concern that can be addressed by making the spacecraft comfortable and trusting the astronauts to be resilient. It is a hard operational problem with real mission failure modes. A psychologically compromised crew makes worse decisions. A crew that has experienced a serious destabilizing conflict operates less effectively. A crew member experiencing clinical depression or a psychotic episode is not just suffering personally. They are a safety risk to the mission and to their crewmates. The work being done to address psychological health in deep space is serious, rigorous, and still significantly incomplete. It will require continued investment, research, and honest confrontation with the limitations of what we currently know, before anyone can say with confidence that the psychological challenges of deep space have been adequately prepared for. Across all of the physiological and psychological challenges we've explored, one truth emerges with particular clarity. We are much better at identifying the problems than we are at solving them. The biomedical research of the past 2 decades has dramatically expanded our understanding of what space does to the human body. The picture is more complete than it has ever been. And paradoxically, the more complete the picture, the more imposing the challenge appears. This is not a reason for pessimism. It is a consequence of intellectual honesty. In the early years of spaceflight, when missions were short and the data sparse, it was possible to believe that the human body would adapt to space well enough for any practical purpose. The bone loss data from longer Mir missions in the 1990s began to complicate that view. The more comprehensive ISS data of the 2000s and 2010s complicated it further, as ANS, discovered in the 2000s, added a new category of concern that hadn't been on anyone's radar. The twin study results of 2019 showed changes across more systems, more persistently, than the pre-study predictions had suggested. Each expansion of knowledge has added complexity and difficulty to the problem. This is, again, how science works. The first measurement is always an underestimate of the true complexity. The second measurement is more accurate. The third is closer still. We are somewhere in the middle of that process of progressive revelation, working toward a picture that will eventually be complete enough to act on with appropriate confidence. The people who will design the first crewed Mars spacecraft will know considerably more about what space does to human physiology than the designers of the ISS knew. They will design and countermeasures that the ISS designers couldn't have included because the problems those countermeasures address hadn't been discovered yet. They will have better radiation shielding strategies, better exercise hardware, better life support efficiency, better food systems, better psychological support infrastructure, better medical capability, and better pharmacological options for the specific conditions most likely to affect a long-duration crew. Will that be enough? The honest answer is that we don't know. We won't know until the mission has been designed, funded, built, launched, flown, and completed. And the crew has returned and been studied with the same rigor that Scott and Mark Kelly were studied after the twin study. The answer will emerge from the mission itself, and from the human beings who fly it. What the scientists and engineers working on this problem believe, what they are betting their professional lives on, is that the answers to the hard questions are accessible, that the solutions to the hard problems are achievable, and that the accumulating knowledge and technology of the coming decades will close the gap between where we are now and where we need to be. History provides some grounds for that belief. The challenges that seemed most intractable at various points in the history of human spaceflight were solved, often in unexpected ways, often by people who insisted on trying in the face of expert skepticism. The oxygen toxicity problems that nearly killed the early high-altitude pilots were solved. The re-entry heat shield challenge was solved. The lunar rendezvous technique that allowed the moon landing was dismissed by many before it worked. The life support systems on the ISS that keep people alive in space continuously were considered engineering impossibilities before they were engineered. The problems of deep space are harder than any of these. The scale of what the human body endures is greater. The distances are longer. The isolation is deeper. The duration is longer. The stakes are higher. And still, somewhere, someone is working on the solution. Today. Right now, in a laboratory, running an experiment that will become a paper, that will become a concept, that will become a technology, that will eventually become a line item in the design specification of a spacecraft that carries human beings further from their home world than any human being has ever traveled. That process is underway. It has been underway for decades. It will continue because the alternative, accepting that the void is permanently beyond human reach, is not a conclusion that the people who have dedicated their careers to this problem are prepared to accept. The real boundary is not physics. The physics can be worked with. The real boundary is knowledge, what we know and don't know, what we can and can't yet do. And knowledge, unlike the speed of light or the mass of a proton, is not fixed. It grows, it accumulates, it compounds. We know more today than we knew yesterday. We will know more tomorrow than we know today. And one day, not so distant as it might seem from here, someone will stand on the surface of Mars and look up at a point of blue light in the night sky, and know that everything that has ever happened in the history of human civilization happened on that point of light, and feel the weight of that knowledge settle into their bones. And their bones, after months in space, will be somewhat less dense than they were when they left Earth, and they will stand there anyway. That is the story of where we are, and it is a better story than it sometimes appears when we count only the obstacles. The obstacles are real. The journey is worth making, and somewhere between those two truths, the future of human spaceflight is being built. The question of what it actually feels like, physically, sensorially, day to day, to live in a spacecraft on the way to Mars is one that no living human being can answer from experience. We have approximations, we have analogs, we have the accounts of ISS astronauts describing life in microgravity, which is the closest lived experience to deep space transit that exists. But the ISS is not a transit spacecraft. And the psychological reality of knowing that you are 6 hours from Earth in an emergency is entirely different from knowing that you are 6 months from Earth in an emergency. The physical sensations of microgravity take time to adapt to. In the first days, most astronauts experience space sickness, a form of motion sickness driven by the mismatch between what the vestibular system expects and what it receives. The inner ear, which evolved to interpret the world in terms of up and down, suddenly finds that up and down have no meaning. The eyes, which on Earth provide a consistent visual confirmation of what the vestibular system says, now contradict it. Fluid in the semicircular canals of the inner ear moves in response to head motions, but the expected correlations between head motion and visual motion no longer hold. The brain, interpreting this mismatch as a sign that something is very wrong, produces the nausea and disorientation that characterize motion sickness. Space sickness resolves in most people within 2 to 4 days as the brain recalibrates its sensory integration. But the recalibration is never quite complete. Throughout the mission, astronauts remain vulnerable to motion sickness if their visual environment or head movement patterns produce sufficiently unusual vestibular inputs. Tasks that involve rapid head movements, particularly in confined spaces with visual reference points close to the face, tend to provoke recurrences. And when the mission ends and the astronaut returns to gravity, the recalibration process runs in reverse. The brain must relearn to interpret vestibular signals in the context of gravity, and motion sickness can recur. The fluid shift that begins immediately upon entering microgravity produces the familiar puffy-faced, congested appearance of astronauts photographed in space. The sensation associated with this shift is a persistent pressure in the sinuses and behind the eyes, a feeling often described as similar to hanging upside down, which in terms of the fluid dynamics involved, it essentially is. This sensation does not fully adapt away. Astronauts report it throughout long-duration missions. It affects the quality of sleep, makes nasal breathing uncomfortable, and contributes to the blunted sense of taste and smell that makes food less pleasurable. The visual environment in a spacecraft is unusual in ways that contribute to a subtle, chronic perceptual strain. The absence of the visual cues that normally signal depth and distance. The way distant objects are slightly blurred by atmospheric haze on Earth, the way shadows fall consistently downward, the way the horizon defines a consistent plane of reference, creates a visual environment that the human visual system processes differently from anything it encounters on Earth. Astronauts adapt to this, but the adaptation is not entirely costless. Some report that returning to Earth, with its rich depth cues and consistent gravitational frame of reference, feels almost overwhelming in the first days after return. The acoustic environment of a spacecraft is relentless and unnatural. Life support systems run continuously. The fans that circulate air through the carbon dioxide removal equipment, the pumps that move water through the thermal control loops, the compressors that maintain cabin pressure. All of these produce a constant background noise that, on the ISS, typically runs at around 65 to 70 decibels in working areas. This is comparable to the noise level in a moderately loud office or a busy restaurant. It is not damaging at these levels, but it is never absent. There is no silence in a spacecraft. There is no moment anywhere in the vehicle where no mechanical noise is audible. Long-term exposure to continuous noise at this level has documented effects on sleep quality, on cognitive performance, and on psychological state. Astronauts consistently cite noise as one of the most significant quality of life issues on long-duration missions. The hearing protection that is routinely worn during particularly loud operations on the ISS helps for those specific tasks. But the baseline noise level is continuous and unavoidable. On a deep space transit spacecraft, designed to be as light as possible and carrying all of the same life support systems, the acoustic environment may be comparable or worse. The electromagnetic environment inside a spacecraft is also worth noting. Electronic equipment generates electromagnetic fields continuously. The communication systems, the computer networks, the medical monitoring equipment, the navigation systems, all of these emit electromagnetic radiation across various frequency ranges. The cumulative electromagnetic environment inside a spacecraft is quite different from the natural electromagnetic environment of Earth's surface. Some researchers have raised questions about whether chronic exposure to spacecraft electromagnetic environments has physiological effects beyond those attributable to the known hazards already discussed. The data are sparse and inconclusive, but the question has not been definitively closed. There are social rhythms that humans rely on without noticing them, rhythms that deep space travel eliminates. The rhythm of seasons, the gradual shift in light quality and temperature that marks the passage of time and anchors the year's structure, the rhythm of social life, the weekend, the gathering, the shared meal with people who are not your immediate family or co-workers, the rhythm of travel, the sense of physical movement through a varied environment, of arriving somewhere new, of leaving something behind, the rhythm of news, the daily update that connects you to the wider human story, however imperfectly. In a spacecraft bound for Mars, none of these rhythms exist. Every day is identical in its physical environment. The time structure is defined entirely by the mission schedule. The social life consists exclusively of the 6 people in the spacecraft, who are also the co-workers and the authority structure and the entire human world. There is no news from Earth that is current. Everything that arrives has been traveling for 10 to 20 minutes. And by the time a reply could be sent, another 10 to 20 minutes have passed. The world outside has moved on. You haven't. This temporal dislocation, the feeling of being cut off from the present tense of human civilization, is one of the psychological effects of deep space communication delay that gets relatively little attention in the spaceflight medicine literature, perhaps because it is harder to measure than bone density or intracranial pressure. But it is real, and it is described by the people who have thought most carefully about long-duration isolation as one of the subtler but more persistent sources of psychological strain. You are not just far away. You are displaced in time. The news you receive is history. The world you left has continued without you in ways you cannot track. The entertainment and cultural resources available to a deep space crew will be extensive by historical standards. Massive libraries of books, films, music, podcasts, and other media will be stored on the spacecraft's computer systems, accessible anytime. Communication with family members on Earth will be possible. With the delay, virtual reality systems may provide some sense of alternative environment, a beach, a forest, a city, for crew members who need relief from the relentless gray and white of the spacecraft interior. These resources are genuine countermeasures to the psychological strain of isolation, and they will be taken seriously in mission design. But they are not substitutes for the things they simulate. A recording of the ocean is not the ocean. A video call with your child, arriving with a 10-minute delay, and departing with your reply arriving 10 minutes after that, is not the same as being present with your child. A virtual reality environment, however immersive, is recognizably a simulation. The human brain knows the difference between simulation and reality in ways that matter psychologically. The crew members who succeed best in long-duration isolated environments, the ones who maintain function, preserve relationships, and return intact, tend to be people who have found genuine sources of meaning and engagement in the mission itself, not just in the compensation and entertainment available. People who are genuinely curious about the scientific questions being addressed. People who find genuine satisfaction in the technical challenges of operating the spacecraft. People who experience the mere fact of being where they are, farther from Earth than any human being has ever been, as itself a source of meaning that sustains them against the grinding difficulty of the daily reality. That quality, the capacity to locate meaning in the missions that is large enough to sustain the person through the mission, may be the single most important psychological attribute of a deep space crew member, more important than stress tolerance alone, more important than conflict resolution skill alone, more important than any specific technical competency alone. The person who can genuinely believe, on the worst day of the mission, that what they are doing matters enough to justify what it costs. That person has a resource that no amount of training, pharmacology, or comfort technology can fully substitute for. This has been true of every significant human exploration in history. The people who made it through the hardest expeditions, who survived the Antarctic winters and the Pacific voyages and the mountain ascents and the desert crossings, were not simply the most physically capable or the most technically skilled. They were the ones who had found or maintained or rebuilt a sense of purpose large enough to carry them through. The ones for whom the goal was genuinely worth the suffering. The first humans to travel to Mars will face suffering. We can say that with confidence. Not the acute, dramatic suffering of a Hollywood disaster scenario, though that is possible too. The chronic, grinding, systemic suffering of a body operating under sustained assault from an environment it was not designed for, in a social and psychological context that strains every adaptive capacity evolution gave us. The suffering is built into the physics and the biology. It cannot be engineered away, not with current or near-future technology. What can be done, and what is being done in laboratories and design offices and training programs around the world, is to reduce it, support it, prepare for it, and give the people who face it every possible tool for managing it and surviving it and coming home. The rest is up to them. And if history is any guide, that will be enough. Because the thing about the people who choose to go to the most difficult places, who look at the cost honestly, accept it fully, and go anyway, is that they tend to find ways. Not always, not without loss, not without cost, but often enough and consistently enough across the history of human exploration that the pattern holds. People go to hard places and find ways. That is not a guarantee. It is not a prediction. It is an observation about human nature that 200,000 years of existence in difficult environments has made very difficult to argue with. The first crew to travel to Mars will carry with them the accumulated knowledge of everyone who has ever tried to understand what space does to the human body and how to mitigate it. They will carry the work of radiation biologists and vestibular physiologists and microbiome researchers and ophthalmologists and psychiatrists and aerospace engineers and food scientists and life support engineers. They will carry countermeasures and monitoring systems and medical kits and pharmacological interventions and psychological support protocols. They will carry everything that every generation before them has been able to contribute to the answer to the question, how do you keep a human being alive in the void? And they will also carry something that no laboratory can produce and no engineer can design, the will to go. That will, in the end, may be the most important thing they carry. The problems are immense. The solutions are incomplete. The risks are real. And the journey is worth making. That is where we are. That is what this means. And that, perhaps, is enough to go on. Let's go back to the beginning. Let's go back to that shield. The magnetosphere that protects the Earth is generated by the movement of molten iron and nickel in the outer core of the planet, approximately 2,900 kilometers below your feet. That movement generates electric currents, and those currents generate a magnetic field, and that magnetic field extends far out into space, tens of thousands of kilometers in the direction facing away from the sun, compressed somewhat on the sun-facing side, where the solar wind pushes against it. The magnetosphere is not a solid barrier. It is a dynamic, constantly fluctuating electromagnetic structure that breathes and flexes in response to changes in the solar wind. During solar storms, it compresses. During quiet periods, it expands. At the poles, it opens, which is why the auroras glow there, where the particles that would otherwise be deflected can follow the field lines down into the atmosphere. Without this magnetosphere, Earth's surface would receive radiation levels incompatible with complex life. Mars, which lost its global magnetic field billions of years ago as its core cooled and solidified, receives galactic cosmic radiation at levels several times higher than Earth's surface, levels similar to what a crew in deep space would experience. The Martian surface is not just cold and thin-aired, it is irradiated. Any future permanent human presence on Mars will require either deep underground habitats, where meters of rock provide shielding, or advances in radiation protection that we don't yet have. The story of the magnetosphere is, in a way, the story of why life on Earth is possible at all. the circumstances that allowed a planet of the right size, with the right composition, at the right distance from the right type of star, to also develop and maintain a protective magnetic field for 4 billion years, while life evolved from single cells to the complexity that can look up at the stars and wonder. Those circumstances are the subject of the emerging field of planetary habitability research, and they appear to be somewhat rare. Not impossibly rare, but rare enough that the conditions we take for granted here are not guaranteed elsewhere. When we think about sending humans to deep space, to other planets, eventually to other stars, we are contemplating sending organisms that evolved in the shelter of those conditions into environments where those conditions do not exist. We are contemplating moving life from inside the cocoon to outside it, and then keeping it alive long enough to do something meaningful before the outside kills it. That is the deepest framing of the problem. Not the technical framing, though the technical details are important. The deepest framing is biological. We are creatures of a particular place, shaped by a particular set of conditions, attempting to exist in places where those conditions don't hold. Everything we've discussed, the radiation, the bone loss, the vision changes, the immune dysfunction, the psychological strain, is the specific mechanical consequence of that fundamental mismatch. The question is whether the mismatch is permanent or correctable, whether the human body, with sufficient technological augmentation and pharmacological support and engineering protection, can operate effectively in deep space for the durations required for meaningful exploration and eventual settlement. Or whether the mismatch is deep enough, fundamental enough, that no practical intervention will close the gap, and that the future of human presence in space requires not just augmenting the human body, but, over generational timescales, changing it. This is not a mainstream conversation in spaceflight medicine right now. The field is properly focused on the near-term missions and the near-term biology, but the longer arc of the question points toward it. The radiation damage that accumulates from high-energy heavy ions is, at its root, a consequence of having a nervous system made of carbon and hydrogen and oxygen, rather than something more radiation-resistant. The bone loss is a consequence of having evolved a skeleton that uses calcium as its structural matrix and requires gravitational loading to maintain that matrix. The fluid, shift, and sands are consequences of having evolved a cardiovascular and cerebrospinal fluid system optimized for a one-gravity, one-atmosphere environment. If we could change those things, if we could design or evolve or genetically engineer a human physiology that handles radiation differently, that maintains skeletal integrity without gravity, that manages fluid pressure without gravitational assistance, we would have a different conversation. Not a conversation about sending the current human body into an environment that will damage it, but about what it means to be human in an expanded set of environments. That conversation is for future centuries. The conversation for this century is, can we protect the human body we have well enough, and for long enough, to begin to explore the solar system in meaningful ways? Can we get people to Mars and back? Can we establish a research outpost? Can we begin the slow process of learning what it means to be a multi-planetary species? The answer, based on where the science and engineering currently stand, is probably yes for limited durations, with acceptable though significant risk for the right people who have been selected and trained and supported with the best current knowledge. Probably yes with the understanding that the first missions will generate data that the second missions will benefit from. Probably yes with the understanding that some cost to the health of the people who go is unavoidable, and that those people will go anyway because they have decided the mission is worth the cost. That is where we are. A probably yes with significant caveats and ongoing work. A probably yes that will become a more confident yes as the research continues and the countermeasures improve and the mission designs mature. A probably yes that is good enough for people with the right qualities, to constitute a reason to go. The universe is enormous. The Earth is small. The distance between them is measured in light years and in the gap between what we can currently do and what we will need to be able to do. But distance, in the history of the human species, has never been a permanent reason not to go. It has been a reason to prepare better and to go anyway. That preparation is underway. The going is coming, and when it does, the people who made it possible will be the scientists and engineers and physicians and psychologists who spent careers on problems that seemed impossibly hard and solved them one at a time, piece by piece, year by year, because the alternative was not going. And the people who will actually make the journey will be the ones who looked at everything we've discussed in this film. Every radiation curve, and every bone density measurement, and every intracranial pressure finding, and every psychological risk factor, and said yes. Worth it. We go. That's the whole story. And it isn't over yet.